Today we are going to discuss about a new spectroscopic technique that is called infrared spectroscopy. Infrared spectroscopy is part of these the advanced chemical spectroscopic techniques which are used to determine different kinds of chemical information, starting from the electronic transition to the vibrational spectroscopy to even the nuclear transitions. So, this normally used in the machines as a Fourier transform infrared spectroscopy or something known as FTIR. So, FTIR is the major Fourier transform technique which is normally used in the experiments, and whenever you go to any research labs they will talk about FTIR. The full form of FTIR is Fourier transformed infrared spectroscopy. Basically it is an infrared spectroscopy, but the informations which are obtained from the machine are basically plotted in this Fourier space instead of the real space, and Fourier space is nothing but the wave number space. Now, this spectroscopic technique, rather infrared spectroscopic techniques, it deals with the infrared region of the electromagnetic spectrum, and I will first give you an idea what is infrared radiation is. Now, as you know, infrared radiation lies between visible and the microwave portions of the electromagnetic spectrum. In the very first class I just showed you the detailed infrared the detailed spectrum of the electromagnetic radiation, where starting from X-rays or gamma rays to radio waves are shown, and rather UV and visible comes in the middle of the spectrum. So, infrared which comes between the visible and the microwave, that is this one here. So, you can see this is the part visible microwave. We have already discussed about the UV and the visible spectroscopy in the last class. So, here the wavelength comes basically falls between these two the spectrums. Infrareds normally have higher wavelengths than the visibles, as clear wavelengths increases in this direction. So, lambda increases in this direction, and the frequency of it decreases as you go from UV to microwave radio. Infrared radiations in region are basically divided into three parts. One is mid infrared, that is exactly the infrared radiation region, and then you have a near infrared and the far infrared. Near infrared means the part of the infrared spectrum that is closest to the visible light, and far infrared means the part which is closer to the microwave regions, and mid infrared obviously is between these two or the middle of that. Primary source of this infrared radiation is thermal, which I will discuss in the next slide. That is basically heat. Now, this radiation is produced by the motion of atoms and molecules in an object. Higher the temperature, more the atoms and molecules move, and more the infrared radiation is. So, any objects radiates radiation in the infrared regions. Even an ice cube actually radiates infrared radiations. So, to give an idea, humans at even normal body temperatures radiate most strongly in the infrared region. That is why there are cameras developed to get images in the infrared spectrum, and these cameras are very popular for the night photography. And you know, this most of the human beings at normal body temperature radiate strongly in the infrared region at a wavelength about 10 microns, and in the image which is shown here in the left part of this figure is the basically the the areas which are red and yellow are these highest temperatures regions, as you can see that. On the other hand, this right side picture shows and night image of a cat using infrared radiations. Yellow white areas are the warmest. So, yellow white areas eyes actually are at a very high radiation coming from the eyes, and the purple regions are the coldest. So, these kind of imaging techniques are widely used for the photographies of in the night, or even sometime in case of the other things. So, I am going to show you the actual values of the infrared radiation, wavelengths and the wave numbers. So, wavelengths falls in the region of 2.5 to 17 micron. That means they are quite large even in compared to the visible. Visibles ones are actually 400 to 800 nanometers. So, if you multiply this one with nanometers, that means if you multiply with 10 to the power 3, so it is basically 200 50 to 1700 nanometers. So, sorry, 2500 to 17,000 nanometers, that is much higher than the visible. And so corresponding wave numbers are basically 4000 to 600 centimeter inverse. These frequencies match frequencies of the covalent bonds basically in the material or in the molecules rather, stretching and the bending vibrations. So you have a covalent bond like carbon-carbon or carbon-hydrogen or carbon-oxygen covalent bonds. There these frequencies actually match very well with the stretching and the bending vibration of those bonds. So therefore one can determine the kind of bonds present inside a covalent bonded material. This is why it is very widely used for covalent bond molecules. So infrared radiations or infrared spectroscopy is normally used to tell you what type of bonds are present in the material and also some type of other structural informations, which we will show you as we go along. So to give you an idea where does it lie, as I said you, this lies between visible and the microwave. The frequencies actually falls in the region of 10 to the power 14 to 10 to the power 12 Hertz, and wavelengths are about 10 to the power 4 to 10 to the power 6 or rather 10 to the power 3 to 10 to the power 5 nanometers. That is what we have seen. And so therefore they talks about the molecular vibrations, and these are can be studied. We have already discussed about UV visible, which can be used for valence electronic transitions, and so IR is basically a complementary technique as compared to the UV visible. Now to give better perspectives of this, infrared spectroscopic process actually deals with quantum mechanical energy levels which are similar to the molecular vibrations. So I do not have time rather to go into the quantum mechanical treatment of the molecular structure. I hope you have already studied in your different courses like physics of metals or maybe some other quantum mechanical courses. So I understand that these techniques are basically this quantum mechanical treatment of molecules gives you the idea of the different quantum mechanical energies, and we know that there are different quantum numbers starting from principal quantum numbers to magnetic quantum numbers to azimuthal quantum numbers to spin quantum numbers. They indicate the different energy levels. So and as in this case actually quantum mechanical energy levels which are used are rather in IR spectroscopy are basically similar to the molecular vibrations. Now we perceive this vibration in terms of heats actually. So that means some kind of heat. That is why I showed you some pictures few minutes back where we can image the animals or even human using infrared camera. When we say a covalent bond between two atoms is of certain lengths, we are citing an average because the bond behaves as there is a vibrating spring connecting between atoms. Let us see the picture here. I have a big atom, there is a small atom. The bond between these two actually can be thought of is basically like a spring or vibrating spring which is connecting them. So that means molecules are vibrating in a certain manner, atoms are vibrating in a molecule certain manner. For a simple molecule is a diameter molecule, we can very easily model this kind of situations using this picture. So if that is the case, that means we know that for any spring there is a vibration this frequency which is very unique to the spring, and if we know the vibration frequency we can know the energy, and if once you know the energy we can clearly say that if we use the infrared radiations we can energize this spring to the higher energy level, and when it comes back to the ground level it will emit the radiation which is absorbed, and by measuring that we can determine type of bond presence in the molecule in the material. So now as I said, bonds between the atoms in a molecule can stretch, can bend when it absorbs infrared energy, and that is how actually the infrared the whole plot between the infrared between the absorption or the transmissions versus wavelength can be prepared here. So to give you some idea, this is called systematic stretch. I will show you some even nice videos. Systematic stretch means you have suppose oxygen, there are two hydrogen atoms, this is like water molecule. So there will be systematic stretch in these two bonds oxygen hydrogen bonds. You can have even asymmetric stretch due to application of the infrared like this, these two are different directions of arrows, or you can have bend, that means you can have basically the hydrogen atoms actually can move little bit or rather vibrate little bit this way, and then that can create a bend. So molecules such as water actually can absorb infrared light when vibration results in a molecular dipole moment change. This is very important as far as the infrared spectroscopy is concerned, because that is how we can actually utilize this technique to determine what kind of particular vibration is present even in the water molecule itself. To give you a better idea, let us consider any kind of atomic energy levels in the atom. These are the ground states or ground vibrational states. You can clearly see these are the atoms, blue color atoms are stating there, and these are the excited states. Now we apply certain wavelengths of infrared energy. What is going to do? These atoms which are present here, they will absorb this, okay, and when they absorb, they go to the excited state. Suppose they vibrate from mu0 to mu1, or even if you apply more energies from mu1 to mu2, okay. So as the time goes on, these atoms which are excited to the mu1 frequency regime can come back to the ground state and leaving behind infrared radiations. If you pump in more energy, these atoms can go into these energy states, high energy excited energy states, and even then they can come back. So when they will come back, there will be a huge change of energy, and that energy will come as a radiations, and that is what has been written here. Absorption basically occurs among the ground vibrational states in the energy difference and the corresponding spectrum determined by the specific molecular vibrations. That is what is important. The infrared absorption is net energy gain for the molecule and recorded as energy loss for the analysis beam. So molecule absorb energy, gain the energy because it is absorbing from the infrared input radiation, and as a result of which this energy loss in the analysis beam, that means the beam which is coming out from the material after interaction there will be energy loss. That can be determined easily. Yes, as I said, I will show you some very nice videos. So as you can clearly see, there are different types of stretch and bends. Stretch, this is all taken from Charles and Abrams, which is this book, classic book by these two authors. As you can clearly see, the stretch is basically symmetric, where atoms can symmetrically move. This is, let me put you, this is suppose carbon atoms and there are 4 hydrogen atoms, and I am just putting 2 hydrogen atoms there and here, and they are moving symmetrically. So that is a symmetric stretch. This is one kind of vibrations, and this vibration will lead to certain kind of energy, and vibration frequency also will be defined, that is mu0, mu1, mu3, mu2 I have shown you. And if you suppose these two bonds are moving opposite to each other at a particular time, like when this one is moving out, this one is moving in, so because of that there will be asymmetric vibrations, that is also possible. There are different states of energies. Okay, so whenever infrared radiation falls on it, it can modify this kind of stretch vibration along the line of the bond. Next one, it can modify the bend. Now you can have a different kind of bends. The one which is shown as is basically in plane bend. It can be either scissor type or rocking type. Scissor type means, suppose in this case both the hydrogen atoms are moving in the same directions, so therefore there is a scissor type. Is scissor, both the scissor, whenever you use a scissor to cut certain things, both these scissor pieces, they come and meet at the going the same directions. Other one is called rocking. Rocking means they are actually moving in the same opposite direction. Sorry, in scissor case these two atoms are moving in the opposite directions, not in the same direction. That is why they can come in scissor, actually they come in opposite directions. In a rocking case, these two atoms are moving in the opposite directions. These two opposite atoms are moving in the same directions. Because they are moving in the same direction, there is a rocking aspects, and this happens when the atoms are moving in the same plane. If the atoms are moving out of the plane, so the first situations in this case like scissor, which correspond to twist. Okay, that they will be twisting the bonds. Second one is called wag, that is both the atoms are moving in the same directions, so that will lead to wag. So you can clearly see that these are all shown here, and scissor is this like this, and rocking is like this, and this is I think twist. So these situations will lead to different kinds of energy states of the molecule. That is what I am trying to impress you upon, so that you can actually use this kind of energy levels to detect the kind of vibration is present in the molecule. That is why it is called vibrational spectroscopy. IR spectroscopy is always known as a vibrational spectroscopy. To give you in better perspectives, IR spectroscopy process actually, as a covalent bond oscillates due to oscillation of the dipole of the molecule. Okay, so these two are actually dipole. A varying electromagnetic field is produced, and because of that, you can see there is a propagation of the radiation, and greater the dipole moment, change to the vibration, and the more intense will be the electromagnetic field generated, and electromagnetic field means there is vibration. So that is what is normally basically classically can be used. Now when a wave of infrared light encounters any oscillating electromagnetic field generated by this dipole of same frequency, the two waves can couple, and then when the two waves couple, that means the IR radiation which is coming and the electromagnetic field which is generated by these oscillating dipoles, they can couple, and when they couple actually this coupled wave can vibrate with the double the amplitude of the initial wave. That is what is shown here. So you can see there this is the input, and this is the because of the electromagnetic field, and when they couple you have electromagnetic wave which is very double the amplitude. So this all can be used. So this is again same things which I told you. I am just going to rush you through this. So these are all vibrations. Vibration means stretching of bonds, bending of bonds, internal rotations, which I have shown you already, and then vibration can change the dipole moment. I have also showed you the asymmetrical stretching or bending, or internal rotations, this wag can change the dipole moment of molecule. Symmetrical stretching or bending does not normally, so they are not active to the IR radiation. Now normally what is the kind of vibration level? So wavelengths I have already told you, wavelengths should be of the order of 2500 to 17 or 15 to 17,000 nanometers. Okay, now vibrations, if you have vibration of suppose 4111 centimeters inverse, that is in stage case of H2 molecule. So can you imagine how many vibrations can be done in per second? It is very large. It can be of the order of 120 to the power 10 to the power 12, 120 into 10 to the power 12 vibration per second. That means one vibration per 10 to the power 15 seconds, which is sometimes is very difficult to detect. That is why you have autosecond laser spectroscopy, where you can use infrared laser, and using autosecond laser spectroscopy you can determine each of these vibration. Autosecond means minus 18 second. So the spectroscopy which is done at the time scale can be used to detect this kind of vibrations, which is now a routine nowadays. Many labs in this, even our country, has this kind of spectroscopies. So this is a very large spectroscopy, and now I have not told you about one thing. This is fine, this vibration, stretching, everything is very fundamental to the IR spectroscopy. This actually gets influenced by the mass of the molecule. To give you some idea, suppose this is a hydrogen molecule, this is the iodine molecule. Hydrogen molecule molecular weight is 2 grams per mole, and for the iodine is 254 grams per mole, huge molecule mass compared to the hydrogen. Now if you look at it, the idea is get the mass, lower the wave number for the vibrations to happen. This is also very important, you must try to remember this. So how much is the movement occurs in a vibration of this CC point now? Let us look at even this nitty-gritty subdetails of that. If I consider this, suppose this is the one atom, carbon atom, this is another atom. Okay. So if they vibrate, approximately what is the length to which it can vibrate? That's distance, actually distance is approximately 10 picometers, and the bond length is about 154 picometers. That's what happens. So for a CC bond with a bond length of 154 picometers, 1 picometer is 10 to the power minus 12 meter, because we know 1 nanometer is 10 to the power minus 9 meter. So that means if the bond distance is or length is about 154 picometers, so that means it's equal to almost like 0.15 nanometers. The vibration is even much lower, it's about 10 picometers. So that is the distance scale I am talking about it. So that is for the stretching. For the bending, what is the bending angle? Well, if we consider the carbon-carbon bonds, bending angle is maximum 4 degrees. So this much of bend happens when the molecule, when the carbon-carbon bonds actually vibrates, bends 4 degrees, and that means distance about 10 picometers. For CCC bonds, angle is changes about 4 degrees, is very difficult, and this moves the carbon atom about 10 picometers. So I will not discuss about these aspects, because I have already discussed with you about the energy and the wave number and other things. So let us look at even a big molecule like pentane. Okay, pentane is C5H10. Okay, so there are 5 carbon atom, 1, 2, 3, 4, 5, and then there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 hydrogen atoms. So if this is the case, then what kind of type of vibration would occur in these big molecules? For small molecules it's easy. Okay, examine this. That can be done using by taking the IR spectrum. This is pentane liquid, and it is measured using FTIR. You can see this is the absorptions, this is the wave number, and this is absorption A plotted here. So you can see the peaks coming in this picture, there are background and peaks coming in the picture, certain wave numbers. There are small peaks, there are big peaks. Okay, and by looking at these peak positions, one can actually easily determine that kind of questions which I have already asked, that vibrations, what kind of vibrations will take place in this molecule. So increasing the absorption of IR radiation, that means as you go 0.2 to 1 absorptions, what happens? CH, this is basically CH stretching, and this is for CH stretching, and this is for CH bending, to separate things, because as you have seen here, so I like to go back, we have carbon, so this is hydrogen, this is carbon, this is suppose carbon, this is carbon hydrogen bond, and this is carbon carbon bonds. That is the overall picture in a pentane molecule. So this is saturated what's called saturated polymer, okay, or alkane rather, and so therefore if I look at it, this is CH stretching, this particular bonds which are coming, the CH stretching, and this is CH bending, and that is what we can clearly see in case of pentane. Now if you look at chloroform and even deuterochloroform, chloroform is very common molecule, this is taken from this source. As you can see, the chloroform is basically shown in the red, and deuterochloroform is shown in blue. So there are certain frequencies or the vibrations we can see. So what you can do is that in a chloroform, chloroform is nothing but CHCl3, that means there are 1 H and 3 chlorine bonds, CCl, CCl, CH, there are 2 types of bonds. So CH stretching for chloroform comes around 3002, this is almost like this, okay, this part, and CH bending comes 1120, so that is this, and CCl comes around 700, 1900, 800, 700, yeah, 700, and CCl bending comes 418, somewhere here comes, sorry, here it can be detected. Similarly for, and these are the measure values, these are normally detected, we can see these things, and for CDCl3, that is deuterochloroform, okay, these are the values, and this we detect all of them. So therefore one can actually clearly say from these measurements what kind of vibrational states of the bonds present in this case. The peak shift is because of H2D, and that is because of mass increase. So you can see that H2D, because of that this peak has shifted, this peak has shifted this. That is what we discussed. The mass increase means more mass means the peaks will come at a higher wave numbers, so lower wave numbers, not higher. Some more results. So calculated values normally in these cases had to be compared with computational values, which are normally done using computational tools. So calculated values, as I showed you here in this case, calculated values are this, and these are the measure values. So there is little bit of variations, but still one can get these results very close to it from the measure value with the calculated value using better computational tools nowadays, which is done. And second thing is increasing the mass will shift the wave numbers to lower values, that is what I told you. And third one, stretching energies are always higher than the bending energies, that we also learned. And bending energies will be always higher than the internal rotational energies, which can occur actually higher wavelengths, which is normally observed. So therefore, does the stretching relationship, next question you have to ask is, does the stretching relation energy have any relationship with the strength of the bond? Actually it is true, indeed true. If you look at wave number versus bond energies, so bond energy is starting from 200 to about 600 kilojoule per mole, wave numbers 0 to 5000. As you can see that as the bond energy increases, wave number also increases. So that means stretching energy is related to the strength of the bond. As the bond increases, the stretching energy also increases. That is what this what's called plot actually suggesting. That is what is the case. So we can plot it and we can get a very reasonable numbers of the confidence using R square. Well, now we can actually go on looking at different kinds of molecules and do that. Most of them will be, today's lecture will be of carbon type. This is formaldehyde, formaldehyde is CHCl2, okay, and this is phosgene, and this is acetone, we know that. And so we will examine this carbonyl groups here. So how does this CO bonds, CO double bonds here, sorry, this one, everywhere you have a CO double bond, okay, everywhere you have a CO double bond, that is in all the cases. So how does this CO stretching energy compare with these molecules? Here actually its wave number is 2053, in case of phosgene it's basically 1951, in case of acetone it's even higher, 2063 centimeter inverse. So that means depending on the structure, formaldehyde, phosgene or acetone, the CO bond stretching energy will change. That's understandable because the environment is changing. The type of atoms present in near these CO bonds, as it changes, the stretching energy also changes. So carbonyl group normally has energies between 1700 to 3000 centimeter. Now one can actually analyze different functional groups in organic compounds, like unlike atomic spectroscopy where the sharp energy transition occur due to well contrasted electron transitions, molecular spectroscopy tends to show bands. We have seen that different kinds of stretching bands. Molecular vibrations are normally influenced by these surrounding groups. We have seen one example in the last slides. So let me just tell you in a brief what we can understand from this kind of spectroscopy. This is stretching and bending energies, and these are the wave numbers. So for the CH stretching energy wave number comes around 3000, but bending will come around 1000 to a couple of hundreds. OH, that is in well groups, the stretching energy can span from 4000 to about 2000, whereas bending energy can span from about 1000, 2000, 1500 to about 500. and then CO is CO means C double bond O, that is the formaldehyde, acetone, all this molecule has this kind of bonds. There the stretching energy can span from 3000 to about 2500, and your what's called the another type of the CO actually single bond can happen, actually the stretching can happen even at lower wave number, that is 1200 to about 1000, and in these cases there are other things like CC double bonds, this alkenes, and it can happen in the range of about very close range about 1500, and then we can actually also have CC single bonds, and this can happen even at much lower wave numbers, that is close to 1000. So using the IR spectroscopy actually one can determine these kind of bonds. Let us see that what else we can do. So therefore we can actually identify different functional groups in the molecule. That is very important to organic chemistry, because you need to know what kind of functional group is present in the molecule which is produced by the chemical reactions, and then only you can basically formulate the structure of the molecule. Special matching can be done by computer software, as I told you, even library spectroscopic data are also available. Since this absorption follows Beer's law, we can actually do quantitative analysis also. I have already discussed to you about Beer's law in first lecture, even in case of UV visible spectroscopy also, how to apply that, and one can actually use this law and then do quantitative analysis also. To do that, let us see that, okay, and this is basically another kind of representation, and I have told all these things already to you, but let me reiterate. Each stretching and bending vibrations actually occurs as very specific characteristic frequencies, which we have already told you, and as you can see that these are actually where the field of electromagnetic field of oscillations interacts with the IR lights. So transmission will be very low. This is basically transmitted is important here, not the absorptions versus wave number, and region where these acceleration bonds interact with light, the transmission is almost like 100%. This is the ones. So that means there is no oscillations of bonds, so there is no transmissions. This is again another kind of plots for transmissions versus wave numbers, and we can see different bands, stretching and the bending bands. We can determine that for different molecules. And to give you some more ideas about it, normally IR spectrums uses unit rather than wavelengths, because wave numbers are directly proportional to the energies, as we have told, because A is nu is the energy, and this can be written as hc by lambda. So hc nu bar is the energy, say where nu bar is basically the wave number. So wave number is directly proportional to the energy, not the wavelength. Wave number is inversely proportional to the energy, that is why we plot it. Higher frequency, higher numbers are equate the higher energy, so short wavelengths equate the higher energy. That is why we use the wave numbers, and wave numbers are extensively used. I do not want to tell detail about that. So let me just, to give you some more idea, it is important to note that peak intensities actually tend to be effects of the three factors. One, if the peak intensity is very strong, that means peak is very tall and absorption is high, transmission is low. Medium ones, which the peak is mid height, that is not very tall and not very small also, so in which this transmission is very small, or you can have a weak, and that means transmission is very high. So if I go back to this plot, I can show you there, this is weak, this is tall, and in some cases you can see medium height here. So all these signals are available everywhere. Sorry. And broad, it can be broad also, then if the Gaussian distribution is abnormally broad, this means actually more of the descending bond, the spans have more energies. Exact transmitted values are rarely recorded here. So that is why, although we can apply the Beer's law, but it is not good to use the exact transmittance to get good results or the quantitative results from that. Okay, so what else the infrared spectroscopy can do? It can actually identify and quantify organic solids, liquid gases. It can analyze powders, solids, gels, emulsions, paste, even pure liquids or solutions, polymers, and even pure and mixed gases. So that means it can analyze everything, solid, liquid, gas. So that is why the versatility of this technique so important. It can be used for all kinds of research activities like quality control, quality assurance, even nowadays we actually as a scientist we use to detect different kinds of bonds presence. Samples sizes can be single fibers like only 20 microns in length to even atmospheric pollutants. Nowadays people do lot of study on atmospheric pollutants, so which can be large. Not only that, it can be used in pharmaceutical research, forensic investigations, polymeric analysis which I have showed you, lubricant formulations and fuel additives. Lubricants which are used nowadays many for the many applications in the machine industry can be studied. Fuel additives, in different fuels we add different kind of additives, and whether they are stable in the fuel conditions can be studied that by this way. Food research is extensively used because food uses a lot of different kinds of spices where this can be used, they are chemicals actually. Quality assurance which I have told you already. Environmental pollutions and the water pollutions can be studied by this way. Not only that, biochemical research or biomedical research can be done. Coatings, surfactants can be studied, they are all even either polymeric or even metallic or the ceramic materials, many others. Most importantly, they are used in the polymers. Well, to give you all these things in a nutshell, that in general the primary use of air spectroscopy is detect the functional groups, but it can be used because it leads to interaction of the electromagnetic spectrum with the actual bonds. It can provide a qualitative probe of the functionality of the molecule because it can actually give different configurations, it can actually give you different configuration of the molecules present in the material, because one molecule can stretch, bend, or maybe you can have different kind of positions or big energy levels, so that can be detected. Since most types of the bonds in covalent molecules have roughly the same energies like CC double bond or CO double bonds or CH or NH single bonds, they show up in similar regions of the IR spectrum. And you must remember that all the organic functional groups are made of multiple bonds, therefore show up in the multiple IR bands. That is why you see a band, that means many peaks are there in one bands. And in general, so we can actually determine 4 regions: OH, NH, CH single bonds, they follow in this range. Triple bonds CC, CN, acetylene, and the other carbon nitrogen bonds 2005, 700, 2000. Double bonds falls 2000 to 1600, and single bonds comes in the small wave numbers, they have the higher energy, so that is CC, CN or CO, they come in the fingerprint regions. So that is why we use these techniques to determine all kinds of bonds.